The second type of membrane transport to look at is facilitated diffusion. This is a second form of passive transport, and passive transport means transport across the membrane that doesn't require an input of energy, or passage across the membrane by diffusion, so from an area of greater concentration to an area of lower concentration. Last lecture I described simple diffusion, which is diffusion across the phospholipid bilayer without the assistance of proteins. Facilitated diffusion is also passive transport and also based on diffusion, but it is facilitated by the action of proteins. Remember that while there are some substances that the membrane is completely permeable to, there are others that still have to cross the membrane but cannot do it by simple diffusion. Those are allowed to move across the membrane still by diffusion, but with the aid of proteins. Remember, I described proteins to you before as they moved through the membrane or were embedded in the membrane. I wanted to go in a little bit more detail with you about those proteins. First, the word amphipathic means that they have both hydrophilic and hydrophobic regions. So here's a polypeptide, a chain of amino acids, at least the first three-letter abbreviations of those amino acids. Those in yellow are hydrophobic. Those in green are hydrophilic. Membrane-spanning proteins are one of the characteristics of membrane-spanning proteins is having this stretch of hydrophobic amino acids that can interact with the hydrophobic barrier. I had described earlier that a lot of these membrane-spanning domains or regions of a protein were helices, and that's what you see here. This helix has R groups that are nonpolar sticking out, and that allows this to be anchored in the hydrophobic barrier, or the hydrophobic stretch of the membrane. Proteins that facilitate diffusion are integral membrane proteins, those that are embedded in the membrane. They often have peripheral membrane proteins attached, but those that actually allow complete passage through the membrane are integral membrane proteins. So some examples of these types of proteins. Channel proteins. Channel proteins allow passive transport of ions. So passive transport, of course, means diffusion. So imagine we've got like a bunch of sodium ions out here in greater concentration, sodium ions in here in lesser concentration or lower concentration. This channel is just the right size and also charge to attract these either molecules or ions. So let's say if these are sodium ions, maybe we have negatively charged amino acids lining this channel. So of course the amino acids that are interacting with the membrane are nonpolar, but you could imagine in this aqueous-filled channel, water-filled channel, we've got some kind of polar or some kind of hydrophilic amino acid. And if the goal is to move positively charged ions through the channel, then negatively charged amino acids will be lining the channel. The ions enter the cell based on the concentration gradient. Greater concentration outside of the cell, lower concentration inside of the cell. Of course, if it was the opposite, if we had a higher concentration here, we would have the solutes or these ions moving out. A second type of protein are the carrier proteins. These aren't channels, but they change shape in response to the binding of whatever the substance is that needs to move across the membrane. For example, glucose. Imagine that these are the cells that are this is a cell that's lining your intestines. And in the lumen of the intestines, just waiting to be absorbed, is glucose. So you've eaten a high sugar meal, the digestive system has broken it down into some monomers of glucose. This is the first layer of cells that glucose encounters. There's more glucose outside of the cell than inside of the cell. There's a protein called the GLUT protein, and it has a binding site for glucose. When the concentration is lower inside than outside, that binding site is facing the outside of the cell. Glucose binds to the protein, and as soon as it does, that binding makes the protein change shape. That's called a conformational change. The conformational change is one where this gets trapped in the protein and the protein opens up at the bottom to release the glucose. Now, as the concentration builds here, this part of the protein actually remains open. So imagine like 10 of these across the membrane of this cell. And as the glucose is being grabbed and brought into the cell, remember that the concentration gradient gets lower and lower, and we start to reach equilibrium, where glucose is the same on one side as it is on the other side. Once the concentration of glucose is equal on both sides of the membrane, you could imagine like 5 of those glucose transport proteins open in one direction, 5 of them open in the other. So it's still a process of diffusion. As the concentration gets higher out here, more of these GLUT proteins open up their binding site. As the concentration gets higher inside of the cell, then we have fewer of these open and more of them with the binding site closed and the exit site open. Now, the opposite doesn't happen with this particular type of transporter. Cells don't actually send, these types of cells wouldn't actually send glucose out because we don't need it back into the intestines. We need to absorb it and do something with it. But there are plenty of proteins, transport proteins, where that is the case, where there needs to be an equilibrium. And so the two conformations are sort of equal as far as bringing solutes in and taking solutes out of the cell. So in facilitated diffusion, we're still moving substances down the concentration gradient, but rather than this movement occurring right across the membrane, it actually requires the help of proteins that are embedded in the membrane. Those two proteins, channel proteins and carrier proteins. Next, we're going to talk about active transport. And active transport is the movement of substances up the concentration gradient. And it requires energy, requires a lot of work to move things from where they're lower concentration to where they're in greater concentration, so up a concentration gradient. Active transport is next.